Hello, my name is John Englehart, and I'd like to thank the organizers for the invitation to speak. I'm going to be discussing my lab's research using ferret models to both understand stem cell targets in the lung and pancreas for gene editing in CF, as well as using genetic ferret models to study gene editing and gene therapy approaches. Uses my disclosures. We're lucky as a field to have 6 mammalian CF animal models to study cystic fibrosis. All of these contract intestinal disease, and they're very extensive pancreatic, liver, and lung disease. However, the pig and the ferret models are the two only models thus far proven to spontaneously develop lung disease like humans. I'm going to be discussing genetic ferret models, stem cell targets in these models, and using gene-edited ferrets to study this, as well as genetic models to study gene editing in both the lung and the pancreas. We've generated a fair number of ferret models using Cas9 protein guide injection and 4 different types of approaches. The first, mediated by non-homologous end joining to either disrupt a gene target or insert a full gene cassette, or homologous directed repair using to insert human mutations using a DNA segment that's inserted, as well as precise insertions to insert cassettes, or as I'll discuss, pre-mediated drivers. The gene disruption models I've listed here. The exon trap generated by gene insertion and ROSA 26 knock-in models are several. We have the Cree reporter that I'll discuss we use for lineage tracing, whole animal Cree, which we hope in the future will allow for easy disruption of CFTR, and 2 models I won't discuss in detail that include a CFTR activation of the endogenous locus in a cell-specific fashion, or overexpression of CFTR in a cell-specific fashion. We have 2 mutations, the G551D and delta F within ferrets, and I will be discussing 2 Cree drivers that we're using to study stem cell compartments within the lung and the pancreas, and conditional CFTR knockout ferrets generated using the precise gene editing approach. If you're interested in hearing about the FOXI1 and mucin models that are enabling better understanding of CFTR function at the cellular level, please go and hear this session's talk. I think it's important when evaluating species to study cystic fibrosis lung disease that there's a recognition of the differences in cell biology within the lung between various species. In the rat and mice, submucosal glands, or rats and mice, submucosal glands are found within either the proximal trachea or the entire trachea. In humans and ferrets, as well as in pigs and sheep, they're found throughout all the cartilaginous airways. These structures are important for innate immunity. They express abundant CFTR and serous cells, as well as ionocytes, and get pathology in cystic fibrosis, as shown here in these human lung sections. Now, the glandular stem cell compartment also houses reserved stem cells that are precursors to committed basal stem cells at the airway surface. These studies were initially performed in mice, and I'll talk about some that we've done in ferret. Basal cells are primarily thought to be the stem cell precursor in the surface airway epithelium of these cell types, including ionocytes, secretory cells, and ciliated cells. In the submucosal glands, there are 5 distinct cell types, including ionocytes within gland ducts, ductal cells, serous cells, mucus cells, as well as myoepithelial cells. Ductal cells have been thought to be a precursor to basal cells in the surface airway epithelium. In my laboratory, as well as the Tata lab, has studied how Wnt signaling engages following airway injury, a myoepithelial reserve stem cell that has the ability to generate these 8 cell types within submucosal glands, as well as the surface airway epithelium. These cell types express CFTR, goblet, ionocytes, basal cells, as well as serous cells. So we questioned, how does the CF disease state alter this submucosal gland stem cell niche? And this is a question that really can't be addressed in mice since they don't develop lung disease. And what tools can assist the field in developing gene editing approaches for glandular myoepithelial cells? So Xiaoming Liu in my lab created 2 different reporter ferrets, one a Cree reporter that expresses tomato and switches to EGFP in the presence of Cree, and then a ACTA2 Cree driver. ACTA2 is expressed in myoepithelial cells of submucosal glands, and created this using homologous dependent insertion of an internal ribosomal entry site in Cree RT2 into the ACTA2 locus. And so in ferrets that are double transgenic for these 2 alleles, induced with tamoxifen in 8 weeks and harvested at 12 weeks, we see that we efficiently can lineage label glandular myoepithelial cells, as well as these isolated smooth muscle cells, as well as vascular smooth muscle cells within the interstitium. Additionally, labeled in the distal bronchi are smooth muscle and submucosal gland myoepithelial cells that still exist. And then in the bronchiolar airways, labeling is in smooth muscle, not in the surface airway epithelium. And respiratory bronchiolar smooth muscle is also labeled. So we tested whether or not glandular myoepithelial cells contribute to airway repair using a polydocanol injury model, using the double transgenic animals induced with tamoxifen, injured 6 weeks later, and then harvested at 1 week after injury to test whether myoepithelial cells can commit to form surface airway epithelial cells. And indeed they did, as you can see here in the tracheal epithelium. This is fairly short time point post-injury. Seen a little bit of differentiation in the green lineage traced cells. One can see clones in the bronchi that don't express tomato but convert to GFP, basal cells in the distal bronchiolar or bronchi, as well as lineage contribution to luminal cells within the submucosal glands. So glandular myoepithelial cells may be a gene editing target for correction of CFTR in both submucosal glands as well as the surface airway epithelium. And studies evaluating changes in these cellular compartments in the CF lung are ongoing and will tell us how the glandular stem cell niche may be involved in airways disease progression. In collaboration with the Tata lab at Duke, we've been evaluating the phenotype of these glandular myoepithelial stem cells in lineage traced animals, in this case 3 weeks of age, FACS sorted green cells from these dissected airways, and added back tomato cells and performed single cell sequencing using DropSeq. And as you can see, there are a number of specific cell types, glandular myoepithelial cells and basal cells falling out here, both expressing GFP, as well as the mesenchymal components, which likely include muscle as well as vascular smooth muscle. And if one takes this data and projects this in pseudotime trajectory plots, here just plotting the PC plots for EGFP positive cells alone, one sees that there are 3 different classes of myoepithelial cells, and using the UMAP plots on pseudotime, one can see that potentially the route of maturation based on the transcriptional changes that occur here bifurcates into both 2 different subpopulations of glandular myoepithelial cells, with GMEC1 potentially being the primordial stem cell for this compartment. Classification of these cells based on genes that are expressed in these cell types include glandular myoepithelial cells here. These are MEC specific genes, as well as serous cells expressing lysozyme and mucus cells. So multiple glandular myoepithelial subpopulations exist, but we do need more sequencing to provide statistics to the pseudotime lineage analysis, but GMEC1 may be the reserved stem cell compartment. In single cell sequencing of lineage traced GMECs at various time points post-injury, we're investigating to evaluate the gene networks that control lineage commitment, and also differentiation towards full-fledged multipotent basal cell on the airway surface. Another lineage tracing line that we've developed is a KRT7 CRE line. KRT7 labels ducts within submucosal glands, as I mentioned earlier. This is also thought to be a stem cell compartment for surface airway basal cells. But it was originally made to label pancreatic ductal cells, as we see here. Labeling central terminal ductal cells are also called central acinar cells here in the pancreas, showing GFP lineage trace, SOX9 positive central acinar and ductal cells, and the tomato in yellow. Also lineage tracing the intercalated ducts within the pancreas of wild-type animals. And our interest in this lineage trace within the pancreas has to do with the fact that SOX9 positive pods expand in the CF ferret pancreas as the acinar cells are destroyed. In this case, we see acinar cells here in red, cyan here, with SOX9 expression predominantly within the lineage trace. These cells, we hypothesize, lead to bipotent ductal progenitors that incorporate into these ducts and eventually acquire a fate here using fluorescent cytohybridization of NGN3 HNF6 negative cells, which have been shown during development of the pancreas to be endocrine precursors to islets. So KRT7 positive ductal cells and or central acinar cells may be viable targets for gene editing of the CF pancreas. And in this case, there would be a selective pressure for survival of cells that were gene edited. We've also developed G550 ferret model and a number of different surrogate endpoints to evaluate therapies listed here. This model, the G550 model, is favorable because animals can be reared both in utero and postnatally on VX770 to prevent disease progression, and disease progression timed by the withdrawal of VX770 is shown here with airway obstruction with mucus and obstruction of submucosal glands. These models, we believe, are going to be useful in studying the reversibility of disease, which is highly relevant to both gene editing and gene replacement therapies, as well as prophylactic approaches to gene editing, in best case scenario, where you gene edit the target before there's disease and evaluate disease progression, or reversal using gene edit or gene therapy. Now, I will just point out that we have two flavors of these models. One was originally reported in Science Translational Medicine. It's a knock-in that was generated by cloning. There's a neomycin cassette in the intron, which reduces expression by 50%. But we have since created a model that has 100% mRNA expression, clean knock-in using the Cas9 mediated approach, as I discussed earlier. And this model, with in vitro ALI cultures, can recover about 75% of CFTR activity in the presence of VX770 as compared to wild type. Now, in terms of therapeutic endpoints that have been used, we can see here that withdrawal of VX770 leads to progressive colonization of the lung, here shown by bacterial CFU in the bowel. These colonization events are polymicrobial, although we do find Pseudomonas here in pink colonizing the animals. And overall, the CFU load per milligram tissue is shown here in a red dot. We've also evaluated lung function progression of disease following the removal of VX770. You can see here using FEV over FEC indications of airflow obstruction within the lung of these animals following removal of VX770. Proteomics approaches in the lung have been very robust for us and have allowed us to evaluate, for example, in this particular case, we're looking at a ratiometric proteomics of bowel in animals prior to and following removal of VX770. And we can see that neutrophil granule are increased within these bowel lungs, as well as MUC5AC. Disease endpoints include lung disease, cystic fibrosis, things associated with infection and pneumonia. And again, PD have shown favorable functional changes in matched animals removed from VX770 and then placed on VX770 for second measurements. So we have made some advances in rearing G551D animals that I think are relevant to others' use of this particular model. Historically, we had bred heterozygote animals, put the gills on VX770 at E28 to prevent meconium ileus. They're born at E42, and then we gavage these animals at day 21, or orally feed these animals at day 21, and could get 100% survival. We've since determined that males homozygous for the G551D locus maintained on VX770 for life are fertile and can give rise, when bred to gills, to 50% Mendelian segregation of CF animals. And in this case, gills are kept on VX770 for the entire conception process. And VX770 is delivered through the nursing gills in the milk, and later orally once the animals can feed from a nipple. And importantly, these animals maintain pancreatic sufficiency and CFTR function. Another model being evaluated by Edel Evans includes the Delta F508 ferret model. Has shown that in polarized airway epithelial cultures treated with VX770 and 809, she can gain back almost 50% function of CFTR here. Looking at chloride currents, in vivo treatment with VX770 and 809 fully protects from meconium ileus in this particular model. And the survival postnatally when maintained on these drugs is significantly enhanced from those that are untreated. And importantly, animals maintained on VX770 and 809 are free from lung disease, but once withdrawn, they develop airway mucus obstruction. So both the G551D and Delta F ferret models may be useful for testing gene editing strategies. We've also been utilizing ferret models to test mRNA delivery approaches, which are compatible with gene editing. This is in collaboration with Arcturus Therapeutics, using a lunar nanoparticle coupled with mRNA that's tagged with this UNA. We've tested this approach using Cree mRNA in the Rosa TG ferrets to look at conversion of the Cree reporter within the lung and biodistribution of the lunar transfection. And in this case, it was highly effective. Here looking at on-phos in the trachea and in the intralobar airways, seeing GFP conversion. And if you want more information, you can see this poster by Carlos. Lastly, gene editing methods that we've been using involve recombinant AAV, attempting to edit the G551D locus using either homologous directed repair or insertion of two large exons of CFTR using a non-homologous end joining approach. Two different vectors have been tested using the one that expresses the U6 guide and has the same gRNA target within the intron of the Rosa 26 locus to capture splicing of a region excised from an AAV vector that has the identical gRNA target, together with Cas9 expressing vector. And in vitro air-liquid interface cultures from these Rosa TG animals, we can see effective insertion and exon capture of this locus. So in summary, genetic ferret models capable of lineage tracing can aid in the identification of stem cell targets for gene editing. Genetic ferret models like the Rosa TG can be used to assess gene editing tools in vivo. And while not discussed, I think these models will be important for generating vectors capable of targeting stem cell compartments. And lastly, we're hoping that the G551D ferret model and Delta F ferret model will be useful to other laboratories since it's now easy to rear on modulators. Thank you. I'd like to thank all the members of my laboratory that have contributed to this work in collaboration with the Tata Laboratory at Duke and Cal Parak at University of Iowa. And I'd especially like to thank Dr. Chinsen Soon, who is the scientist that does all the embryo manipulation and generation of these new genetic ferret models, among many other things. Thank you.
Hello, and welcome to the 2020 North American Cystic Fibrosis Conference, and thank you for tuning into my talk on our work on doing CFTR, cDNA targeted insertion into the endogenous locus, and our work on understanding the immune response to Cas9 for potential in vivo genome editing. I have been involved with several biotech companies. None of these have had any impact on the academic work I'm going to describe, and none of these companies actually have any FDA-approved drugs that can be prescribed. So I'm going to first talk about the efficient ex vivo gene targeting system that we have developed and now utilize quite broadly, and then describe how we apply that to a gene targeting correction system of the cystic fibrosis transmembrane conductance regulator in primary human airway basal cells, the stem cells of the airway epithelium. And then conclude with part two of the talk about some of the issues of immunity to Cas9 as a barrier to in vivo editing, including intracellular innate immunity, the acquired immunity to Cas9 after it's delivered, and pre-existing immunity to Cas9 among healthy adults. So genome editing, as probably all of you know, now provides a precise method of genetically engineering cells. The power of genome editing was really unleashed with the discovery and then development of the CRISPR-Cas9 system, which was recognized this year with the Nobel Prize in Chemistry to Emmanuel Charpentier and Jennifer Doudna. The system generally works by designing a nuclease to recognize a specific target site in the genome, where it creates a specific double-stranded break. The cell then repairs that double-stranded break either by non-homologous end joining, which can cause insertions and deletions at the site of the break, or by homologous recombination. And if one can provide a repair donor to the cell at the same time it is repairing the break by the recombination pathway, one can create precise changes in the genome down to the single nucleotide level up to the insertion of large cassettes of genes. And it is this pathway that I will focus on the rest of my talk. So the system that we utilize is an ex vivo system in which we purify our cell of interest. We have recognized that the homologous recombination pathway is active in dividing cells, so have to do cell biology to optimize the cell culturing conditions. We then deliver the nuclease as a ribonuclear protein complex in which a purified Cas9 protein, and we use high fidelity versions first discovered by IDT, but now also licensed and sold by Aldevron, complex to what I call an S-cubed guide RNA. S-cubed because it's synthetic, single, and stabilized by end modifications to protect it from nuclease degradation. And we deliver this RNP complex by electroporation. And then we deliver the donor template using the non-integrating adeno-associated virus type serotype 6. And in this way, this virus has evolved ways of delivering its nucleic acid content to the nucleus of cells without being detected. So the concept of using cell engineering or stem cell engineering can be applied in two broad ways. One is to simply rescue a missing genetic function, and that is what I will focus on in the next few minutes, and particularly focus on what this conference is most interested in, which is the CFTR gene, because mutations in this gene are the cause of cystic fibrosis. And recently, a multi-laboratory piece of work was published in Cell Stem Cell, and this multi-lab collaboration was led in an incredibly effective fashion by a postdoc in my lab, Sriram Vaidyanathan, who has just recently received a K99 score that is fundable, and hopefully he will soon be moving on to his own independent position. The idea is that the airway is a very challenging place to think about doing ex vivo transplantation of genetically corrected cells. So the strategy we have taken is not to go to lower airways first, but to access the sinuses, which are a very accessible part of the airway. The sinuses are also a site of significant disease in cystic fibrosis patients. And with our ENT colleague, Jay Karnayak, we're able to acquires basal cells of the sinus, able to then expand them, as I said, under special optimized culture conditions, and then edit them using the RMP-AAV6 system I've described. And of course, unlike the blood system in which we can transfuse hematopoietic stem cells into the bloodstream and they will naturally home to find their niche and reconstitute the blood system, we will be unable to do this with airway basal cells. And instead, our idea is to embed them into a biomaterial that then the cell biomaterial would be what would be transplanted back into the sinus. So of course, as you're all aware, this is an interesting gene because it has most patients, 70% of patients, have a single mutation, a deletion of 3 nucleotides causing the delta F508. But of course, there are thousands of other mutations that also lead to this disease. So in the next few minutes, I will describe our work in both directly correcting the delta F508 mutation and then designing a more universal strategy. So the delta F508 system involves designing a donor nuclease that targets the sequence adjacent to the deletion, and then providing the AAV6 donor that would add back 3 nucleotides. And what we find is that in airway basal cells, the frequency of allele correction ranges between close to 40% to almost 50%, depending on whether we use sinus airway basal cells or bronchial epithelial cells. When those corrected cells are differentiated in air-liquid interface as part of an air-liquid interface culture and conductances are measured, what we find is of course, CF patients have very low conductances relative to wild-type levels, but the frequency, the level of correction of the current is directly proportional to the level of editing. And we can reach levels that are between 20 and 100% of wild-type levels. It is estimated by the physiologists and experts in the field that reaching a level of 15% of wild-type would be sufficient in an epithelium to dramatically change the course of this disease, if not cure it. So what we're doing with this system now is providing a systematic study of different biomaterials for embedding and then transplanting our gene-corrected basal cells in. And then instead of guessing at what the FDA wants is to put the work in to set up an interact meeting with the FDA to have a discussion about what the appropriate safety and efficacy animal studies might be needed before we can move on to initiate a phase 1/2 trial. In parallel to the direct correction of delta F508, Serum has worked on developing, as I said, a universal correction system in which a full cDNA would be inserted into the endogenous locus and thereby correct close to 99% of the mutations that cause cystic fibrosis. The challenge is that the CFTR cDNA is too large to package into AAV6. And so we turned to a discovery that was made by another postdoc in my lab, Rasmus Bach, who now runs his own lab in Denmark. And what he showed quite remarkably is that in a single shot, one can get 2 integration events where a single break would catalyze the integration of donor 1, then that integration would recreate the binding site for the CRISPR-Cas9, create another break, and catalyze the second insertion of AAV of donor 2, and thereby you could expand the capacity of the system. And in hematopoietic stem and progenitor cells and T cells, the frequency of this event could be between 5 and 20%. This would then give us the capacity now to not only insert the full cDNA, but also allow us to insert a promoter with a inert cell surface selection cassette to enrich for those cells. So airway basal cells were not quite as effective as the hematopoietic stem progenitor cells in this double targeting approach with an overall frequency of about 3%. But because we inserted a selection cassette, we could use enrichment strategies, whether flow cytometry based or magnetic bead based, to enrich the frequency of these cells to over 60%. So we then, Suram then, with his collaborators, differentiated these cells, as described previously, on air-liquid interface cultures and measure currents. And what's quite interesting is, here's an example plot or a trace, is that the enriched universal corrected population actually shows currents as that are equivalent or perhaps even greater than wild type levels. And it plotted in a slightly different way than what I showed before is, again, the current in uncorrected cells is quite low. Here's the range of current that is found in non-CF air-liquid interfaces, and you can see our enriched universally corrected cells fall within the range of wild type cells. So this method, of course, requires more culturing in vitro. And so one of the things we looked at was whether the extended time in culture would shorten telomeres and thus perhaps cause a problem with senescence after the cells were transplanted into the patient. And what we find, quite interestingly, is that the basal cells maintain their telomere length for multiple passages, and this was shown across 4 different donors. There is a slight shortening of the telomeres, but there is certainly not significant shortening that would preclude thinking that these could provide a sustained, durable effect if transplanted successfully. So again, we will need to find the successful biomaterial and approach to transplanting the cell biomaterial into patients. So in the second part of the talk, I'm going to talk about the variety of immune responses that we and others have identified to Cas9. And these immune responses, I think, pose a significant but perhaps solvable barrier to doing in vivo gene correction for CF. The first barrier is that cells, unlike the tissue culture cells we use in the lab, like HEK293Ts and HeLAs and K562s, which for reasons we don't understand have inactivated their cytosolic DNA sensing mechanisms, primary human cells have that sensing mechanism intact and thus robustly activate a type 1 interferon response when cytosolic DNA is detected. So this becomes a barrier when one thinks about delivering a donor molecule in a nanoparticle, because it is likely that that donor DNA molecule would be sensed by these airway basal cells and activate a toxic type 1 interferon response. The second type of immune response that we'll have to deal with is the adaptive immune response to Cas9. So in mice that are naive to have seen Cas9 in their lifetimes, because Strep pyogenes and Staph aureus don't routinely infect mice, particularly laboratory mice, Amy Wager's group and Charlie Gersbach's group both found that an adaptive immune response was mounted against the Cas9 that was delivered on an AAV. And in the Wager's group experiments, they found that the T cells, even in these inbred mice, recognized epitopes throughout the Cas9 protein, I think making it quite challenging to think about engineering away this immunogenicity. Similarly, Charlie's group found that adult mice developed robust antibodies to Staph aureus Cas9 delivered on an AAV, although interestingly, they found that when the AAV was delivered to neonates with an immature immune system, the antibodies did not develop, suggesting that there might be a window that one could deliver these genome editing reagents to human beings. And then finally is Karsten Charlesworth, when he was a technician in the lab before he joined the lab of Hiro Nakauchi as a graduate student, took on a sort of not a challenging project, but a project that many in the lab thought wasn't going to work. And as many of you know, the most active Cas9s that we use both in vitro and in vivo is derived from bacteria that commonly colonize or infect human beings. And so the hypothesis that Karsten and I discussed was whether that chronic colonization might lead to an adaptive pre-existing immune response to the Cas9 proteins. And so he developed an ELISA to detect the humoral immune response, and a standard ELISA reaction to detect pre-existing antibodies to Cas9 protein. And he performed various titrations of dilutions of serum to figure out the optimal dilution to use. And what you can see is that as you dilute the serum, the overall signal does decrease, but at low dilutions, the signals are quite high. And so we ended up choosing 1:50 dilution. As our positive control, we used tetanus toxoid, and as our negative control, we used human albumin. What you can see is the population of blood donors at the Stanford Blood Bank are generally immunized. And so expanding this out to 125 different samples, Karsten found that close to 80% of healthy adult blood bank donors had pre-existing antibodies to Staph aureus Cas9, and 60% had pre-existing antibodies to Strep pyogenes Cas9. One might say, well, antibodies aren't the problem because we won't be delivering protein to the serum, and so what about T cells, which could actually recognize cells expressing Cas9 protein when the peptides are expressed on the surface? And so for this, Karsten developed an ELISpot assay. And to cut to the chase, similarly, a large fraction of adult healthy humans have pre-existing T cells that recognize Cas9 epitopes on the surface of cells, suggesting, again, that there's a pre-existing adaptive immune response that might be a significant barrier to delivering Cas9 in vivo to humans. And this was confirmed. These general results in the ELISpot and ELISA were confirmed by a number of different assays and have been generally confirmed by several different groups over the last couple of years. Finally, the group at Rice and Baylor have tested how limiting this might be by modeling the presence of pre-existing immunity by immunizing mice to Cas9, either with purified Cas9 protein or as a control of albumin, and then challenging those mice with AAV delivering the Staph aureus Cas9. And what they found is that the immunization resulted most dramatically here at 12 weeks with the complete loss of genetically engineered cells in the liver of these mice. Although the process was slow enough that the mice actually didn't die, but they just lost the gene-edited cells, presumably from the T cell clearance. And again, T cell infiltration into the liver was demonstrated of the cells still expressing the Cas9. So this highlights that we're going to need to find ways of minimizing the duration of expression of the Cas9 so that the adaptive immune response or pre-existing T cell response will not eliminate those cells. So what are some of the challenges as we move forward in the genome editing space in terms of doing in vivo editing? The first is that the homologous recombination pathway is really only active in cycling cells, and endogenous in situ basal cells are only slowly cycling. So I think systems that allow targeted integration that work in non-dividing cells is going to be necessary. And we must be able to deliver those systems with enough quantity to achieve that 15% gene targeting or gene correction. And the delivery has to occur across, obviously, the thick mucus layer or some way of getting rid of that layer to deliver the editing reagents. All while we avoid the three aspects of the immune response that I just described: the inflammatory response to the cytosolic nucleic acids, the pre-existing and acquired adaptive immune response to the gene editing reagents, and what I didn't mention and discuss is, of course, the pre-existing and acquired adaptive immune response to the delivery vector itself. Nonetheless, as a postdoc in my lab and I have been talking about, just because there are challenges doesn't mean that there aren't going to be solutions with a dedicated, creative, and smart effort. With that, again, I want to thank you for the opportunity to present at this 2020 conference. I really want to highlight Shriram Vaidyanathan for the incredible work he's done on pioneering ex vivo gene correction in primary somatic basal cells as a potential approach to treating or even curing cystic fibrosis. And with that, I will thank you and be happy to take any questions that occur through the chat.
Hello, my name is Alexander Souza, and I am a graduate student in the lab of Dr. David Liu at Harvard University. I'd like to thank the organizers for letting me speak in David's place today. The title of my talk is Base Editing and Prime Editing, Genome Editing Without Double-Strand Breaks. I have no relationships to disclose related to this presentation. Human gene variants that cause genetic disease are diverse. They include transition point mutations, such as the CDT mutation that results in progeria, transversion point mutations, such as the A-to-T mutation that results in sickle cell disease, small insertions, like the 4-nucleotide insertion responsible for Tay-Sachs disease, or small deletions, like the 3-nucleotide deletion responsible for the common F508del mutation that results in cystic fibrosis. A long-standing goal of the genome editing field has been to install or correct all types of pathogenic mutations so that we can study or treat the broadest possible range of corresponding diseases. Programmable nucleases, such as CRISPR-Cas9, have enabled targeted gene disruption by acting as molecular scissors that induce DNA double-strand breaks. These double-strand breaks, or DSBs, result in imprecise insertions and deletions, or indels. While double-strand breaks are useful for disabling a gene, the majority of genetic diseases instead require the precise correction of point mutations, small insertions, or small deletions. Programmable nucleases are capable of installing precise edits through homology-directed repair, or HDR, but this method remains inefficient in certain cell types and is challenged by the need to co-deliver a donor DNA template. To provide one potential solution to the challenge of installing precise DNA edits without double-strand breaks or donor DNA templates, a group developed base editors. Base editors use catalytically impaired CRISPR-Cas nucleases to target a desired DNA sequence, but instead of cutting the DNA, they use naturally occurring or evolved deaminase enzymes to directly convert a targeted base to another base. Additionally, base editors guide DNA repair processes to make this conversion permanent on both DNA strands. The first reported base editors were cytosine base editors, developed by a team led by former postdoc Alexis Comore, now an assistant professor at UCSD. Cytosine base editors are fusions of 3 proteins. First, a catalytically impaired Cas9, shown here in blue, which cannot cut DNA but retains its ability to bind DNA in a guide RNA programmable manner. Second, a cytosine deaminase enzyme, shown here in red, which catalyzes the hydrolytic deamination of cytosine to uracil in single-stranded DNA. Third, a uracil glycosylase inhibitor, shown here in purple, which protects the installed uracil from removal by the cell. The Cas9 domain of the fusion protein binds one strand of the target DNA site and pairs one strand of DNA with the guide RNA. The unpaired DNA strand forms a single-stranded bubble, within which all cytosines falling within an approximately 5 base pair window are deaminated into uracil by the cytosine deaminase domain. The uracil glycosylase inhibitor domain protects the uracils from base excision repair by the cell. At this point, a UG mismatch is present. To buy a cellular mismatch repair to fully convert a CG base pair to a UA base pair, the unedited G-containing strand is nicked by the catalytically impaired Cas9, which is then repaired using the edited uracil-containing strand as a repair template. The final editing product is a UA or TA base pair. The conversion of CG base pairs to TA base pairs by cytosine base editors is able to theoretically correct up to 14% of known pathogenic human SNPs. However, the majority of disease-associated point mutations are corrected instead by converting an AT base pair to a GC base pair, the opposite edit to what a cytosine base editor installs. In 2017, a team led by former postdoc Nicole Godelli, now a director at Beam Therapeutics, developed adenine base editors, or ABEs. ABEs catalyze the hydrolytic deamination of adenosine to inosine, which is read as guanine by polymerases during transcription or DNA replication. ABEs function like CBEs. They bind, deaminate, and nick their target DNA, but instead make edits that convert AT base pairs to GC base pairs. The capabilities of base editors have been expanded by our lab and others since their original development. One recent LULAB story that I would like to highlight is the phage-assisted continuous evolution of ABEs, led by postdoc Michelle Richter and graduate student Kevin Zhao. Using this system, commonly known as PACE, Michelle and Kevin were able to rapidly evolve new ABEs with greatly improved editing activity. They used a selection scheme that required the correction of 2 stop codons in a T7 polymerase gene to permit the replication of phage-encoding evolving ABE variants. ABE variants with the highest editing activity were enriched, and the product of their work was ABE8E, the successor to the originally published ABE 7.10. In collaboration with Jennifer Doudna's group, Michelle and Kevin found that ABE8E is about 600 to 1100 times faster than ABE 7.10. ABE's higher activity enables editing levels in well-transfected cells to approach nearly 100% conversion of AT base pairs to GC base pairs. In addition to its improved activity, the evolved deoxyadenosine deaminase domain of ABE8E overcame limitations of ABE 7.10, namely the incompatibility of the original ABE 7.10 deoxyadenosine deaminase domain with Cas domains other than SpCas9. Michelle and Kevin observed improved ABE8E activity over ABE 7.10 with all of the alternate Cas domains they tested. This included Cas9 homologs, Cas12a variants, and engineered Cas9NG variants, many of which were not compatible with the original ABE 7.10. Michelle and Kevin's work to develop ABE8E has important implications for the LULAB's goals to correct pathogenic variants of the CFTR gene with base editors. Because of ABE8E's greatly improved editing activity and compatibility with many different Cas domains, we now have a new tool to use in our efforts to correct mutations stemming from G to A mutations, such as the gating mutation that results from the G551D variant of the CFTR gene. This specific mutation could be corrected by an ABE8E-mediated A to G base edit. Base editors can correct pathogenic transition mutations. Cytosine base editors can make C to T and G to A edits, and adenine base editors can make A to G and T to C edits. However, there are other pathogenic mutations beyond just transition point mutations. These other mutations account for 70% of known pathogenic gene variants, and the LULAB sought to develop methods to correct or install transversion point mutations, small insertions, and small deletions without requiring double-strand DNA breaks or donor DNA templates. Our group set out to develop a search and replace genome editing method that would not require double-strand breaks nor a donor DNA template. We envisioned using a special extended guide RNA, which we termed a prime editing guide RNA, or pegRNA, which would not only specify the target site, but also encode the desired edit information. We also realized that we would need some way to transfer this edit information to the targeted site without creating a double-strand break or using a donor DNA template. We envisioned that the transfer of this edit would result in a DNA flap on one strand that will be resolved into heteroduplex DNA, and we also recognized the need for a strategy to help the cell copy this edit to the unedited complementary strand. Before I continue, I should make clear that this project was spearheaded and orchestrated by a brilliant postdoc, Andrew Anzalone, and I was lucky enough to work with him on the original prime editing development team. To fulfill the goal of using pegRNAs to specify a target site and also transfer edit information to the target site, we envisioned nicking the targeted DNA with a catalytically impaired Cas9 nuclease. From there, a complementary sequence element present on the 3' extension of the pegRNA would bind the free 3' end of the nicked DNA, thereby priming reverse transcription and leading to the installation of an edit on the nicked DNA. This strategy would require the pegRNA extension to feature 2 elements: a primer binding site complementary to the 3' end of the DNA nick, and a reverse transcription template including the desired edit. Successful priming and reverse transcription from the 3' extension of the pegRNA would lead to the extension of the 3' nicked DNA, resulting in a 3' flap containing the edited DNA. We envisioned that we could include unedited sequence after the edited sequence present within the extended 3' flap, and that this 3' flap might displace and exist in equilibrium with the unedited 5' flap of DNA, shown here on the top strand. Cells treat 5' flaps and 3' flaps differently. 5' flaps are common intermediates in DNA replication and repair, and cells have evolved nucleases like FEN1 or EXO1 that hydrolyze 5' flaps. We envisioned that preferential 5' flap excision would drive the incorporation of the edited 3' flaps to completion, resulting in heteroduplex DNA containing an edited and unedited strand. These strategies were verified after extensive preliminary work in vitro and then in yeast, and we arrived at our first iteration of prime editing for mammalian cells, PE1. The two-part PE1 system used first a fusion protein consisting of an SpCas9 nickase fused at its C-terminal end to an MMLV reverse transcriptase, and second, a prime editing guide RNA, or PEG RNA. Importantly, we discovered that primer binding sequences of lengths greater than 8 nucleotides were necessary to detect prime editing in mammalian cells, and we also observed more efficient editing when the reverse transcriptase was fused to the C-terminal end of the Cas domain rather than the N-terminal end of the Cas domain. Our initial observed editing rates with PE1 were modest, primarily falling in the sub 5% edit installation range. We set out to improve upon PE1 by engineering the MMLV reverse transcriptase domain. We tested multiple variants of this enzyme, each containing combinations of previously reported mutations that improve properties such as template binding or processivity. After screening 19 mutants, we settled on a pentamutant reverse transcriptase that greatly increased the efficiency of prime editing, and called the prime editing system that used this new mutant PE2. Across a handful of sites, we observed that PE2 improved prime editing efficiency 2 to 5-fold over PE1. We also observed that prime editing efficiency could be further optimized by testing multiple combinations of pegRNA primer binding site lengths and pegRNA reverse transcription template lengths. In HEC293Ts, we found that we could use PE2 to install transversion mutations at rates between 10 and 20%. These edits were previously impossible with base editors. The PE2 system provides a method to copy an edited sequence into a 3' flap, and then have this edit resolved by cellular flap resolution into a heteroduplex of mismatched DNA that contains one edited strand and one unedited strand. To guide cellular resolution of this heteroduplex in a manner that favors the edited strand, and also hopefully increase prime editing efficiency beyond the level seen with PE2, we borrowed a strategy originally developed for base editing. We nicked the non-edited strand of heteroduplex DNA, biasing cellular repair to replace the non-edited strand using the edited strand as a template. Because the PE2 system already relies on a Cas9 nickase, introducing an additional nick to target the unedited DNA strand was only a matter of additionally including a standard sgRNA in our prime editing experiments. We found that including this additional nick targeted to the unedited strand increased prime editing levels 2 to 3-fold greater than levels seen with PE2. We also noticed that indels increased with the inclusion of the nicking sgRNA, but noted that these indels remained lower when the non-edited strand nick was kept greater than 40 nucleotides away from the pegRNA-induced nick. With the addition of the nicking sgRNA to the PE2 strategy, we reached our latest iteration of the prime editing system and called it PE3. We found that PE3 offered exceptional versatility in the types of edits that could be introduced in human cells. Here we can see results from our first PE3 characterization experiments, where we demonstrate that PE3 can introduce all 24 possible single base substitutions, insertions, deletions, and combinations of multiple edits. These edits were completed with reasonably good ratios of intended edits to indels, And for the first time, these edits were completed without the need to induce a double-strand break or provide a donor DNA template. It is worth noting that the vast majority of pathogenic insertions and deletions are small. By correcting insertions, deletions, duplications, and indels equal to or shorter than 20 base pairs, prime editing is able to correct the majority of these variants, which represent 39% of known human pathogenic variants. Traditionally, one of the primary constraints of CRISPR-based genome editing methods has been the availability of PAMs near a target of interest. If a target of interest existed in a section of the genome with few compatible PAMs, it would be difficult or impossible to edit. While characterizing PE3, we observed that by increasing the length of the reverse transcription template, edits could be placed increasingly farther away from the pegRNA nick, in some cases, dozens of nucleotides away. This flexibility for performing distant edits demonstrated that prime editing is less constrained by PAM availability than other precision genome editing methods, in addition to being able to introduce a variety of previously unavailable edits. Compared to Cas9 nuclease-initiated HDR, we found that prime editing installs edits with greater or equal efficiency, but with greatly reduced indel levels. Because prime editors do not create double-strand breaks, they display approximately 100-fold higher edit-to-indel ratios than optimized HDR strategies installing equivalent edits. Moreover, prime editors also forgo the need to co-deliver an HDR donor DNA template, which can be challenging and invoke an innate cellular immune response. Prime editing and base editing offer complementary strengths. When a target nucleotide is well placed within a base editor's edit window, base editors offer higher editing efficiency with lower indels than prime editing. This is the case with nucleotide C7 in this plot. However, when a target nucleotide is poorly placed within a base editor's edit window, such as C3 here, prime editors offer higher editing efficiencies than base editors. Additionally, because one can dictate the exact edit outcome with a pegRNA's reverse transcription template, prime editing is more precise than base editing. For example, while a cytosine base editor would indiscriminately edit 3 Cs falling within its edit window, PE3 can specify whether the first, second, or third nucleotide is edited. As such, prime editors offer better precision and targeting scope than base editors. Prime editing strengths are suited for correcting the diverse number of pathogenic mutations that result in cystic fibrosis. Without causing double-strand breaks, nor needing to supply a DNA donor template, prime editing can correct small deletions like that of F508del, transversion mutations like that of G542X, and targets that would otherwise be complicated to correct with base editors due to bystander edits, like W1282X. These CFTR targets and others are the subject of our ongoing cystic fibrosis prime editing research. Today, I've covered recent developments in precision genome editing from the Lu Lab, and how they can be applied to correct pathogenic CFTR mutations. In particular, base editors can install transition edits consisting of C to T, A to G, T to C, and G to A. And prime editing can use a nicked target site and reverse transcription to install any single nucleotide edit, small insertions, and small deletions. Importantly, both of these methods do not require double-strand breaks, nor the co-delivery of a donor DNA template. I would like to acknowledge the funding sources listed here, and also the scientists whose work I have shared today. I would like to thank my thesis advisor, Dr. David R. Liu, and also Dr. Greg Newby from the Lu Lab. Thank you again to the organizers for orchestrating this meeting, and thank you to the audience for listening in today.
Folks, hello, I'm Fyodor Urnov. I'm a professor in the MCB department at UC Berkeley. I work at the Innovative Genomics Institute, and it's a pleasure for me to speak with you and about what I decided to call a lexicon for the age of genome editing. I'll introduce the technology. Many of you are familiar with many of its principles, and throughout the presentation, I will aim to focus on things that are directly relevant to the CF community. People often ask, what's it like at the IGI now that Jennifer has just won the Nobel Prize for developing a method for genome editing. And of course, Jennifer founded and is directing the Institute. And the honest to goodness truth, after an amazing day of celebration, there was a party, and here's this great picture. Things went kind of back to normal, and Jennifer just had her lab meeting and a few days ago, and I always join because it's amazing science. And there she is, just a few days after winning the Nobel Prize, focusing on the science. So it's quite remarkable. So now on to the matter at hand. The Nobel Prize winning genome editing. What's its core vocabulary? We of course start with the genetic material, and one thing I want to point out is the visual uniformity of the double helix is a bit deceptive as far as a genome editor is concerned, because of course not all base pairs in the human genome are made equal. This is the so-called design density problem, and in English that means, can you design an agent that can recognize specifically your sequence? And for cystic, which of course, as you know, is caused by about 1,500 mutations or perhaps more, this creates a really interesting challenge as far as how do you build a relatively broad genome editing or other type of platform that will address those individual mutations. And you'll hear more about some approaches towards this later in the talk. But in the very big picture, it creates a really fascinating challenge for, in particular, individuals with really rare mutations in CFTR that aren't covered by the Vertex medicines, and for which thus you kind of have to build a bespoke gene editing approach, more or less along the lines of what Tim Yu at Children's Boston did for Mila with Batten's disease. I'll talk more about this in a second. One of the really amazing things about genome editing is the way it collaborates with natural processes. The dominant one is double-strand break repair, although, as you'll hear in a second, subsequent developments in the genome editing field have managed to co-opt other DNA repair processes that you see on this screen as well. The one that is the most focus is the one that emerges and gets fired up after you have a double-strand break due to replication fork collapse, or when you take a flight, or you get a chest x-ray or a dental x-ray, or when you have a double-strand break induced by an engineered nuclease. And then there are two pathways that kick in. Now they're actually, the short punchline is it's complicated, but the bigger point is that there are more or less two types of outcomes that we anticipate that are directly relevant to cystic, and I'll touch on both of them. So the agent of change in any genome editing is an engineered protein. They come in many flavors: zinc finger nucleases, tau effector nucleases, meganucleases, and megatals, and of course RNA-guided nucleases, which again just won the Nobel Prize for Jennifer and Emmanuel. I will come back in a little bit to the question of whether or not any one of these is, quote, preferred versus not for the challenge of a gene-based approach, a gene engineering-based approach for CFTR. But I do want to highlight that we live in this remarkable time where, in fact, all of these nuclease classes are currently in the clinic, including, importantly, for zinc fingers and for CRISPR-Cas, they're in the clinic for in vivo editing as well. And this is important for the CF community because this means that there is more or less a charted path for how to put one of these into the clinic in an in vivo setting. Although there's some really interesting thoughts about how to, and experiments, how to use an ex vivo approach for CF as well. And we hear more about this later today. What do you need to know about these nucleases? Well, pretty much only this. They recognize DNA via different principles. Zinc fingers and tau effectors use proteins to recognize the target sequence. Cas9 is an RNA-guided nuclease. But whether you use the first, the second, the third, or some other category, I would like to go back to the point I made on my very first slide, which is DNA may look visually uniform in the beautiful representation of the double helix, but the underlying sequence heterogeneity and the context in which individual CFTR mutations that cause cystic occur really affects how editable they are. And this is just something that everyone should keep in mind as you're approaching the development of a gene editing approach of any flavor for cystic. Now, it's fascinating now that Jennifer and Emmanuel have been honored with a Nobel Prize, which is overwhelmingly well-deserved, that there will be legions and legions of high school students that will have to memorize what CRISPR, the acronym, stands for. And one of the funny things is, to me, is that the acronym has a meaning which is not useful to genome editing, because obviously it starts, clustered regularly interspaced short palindromic repeats. I mean, moving on. But what I think is truly noteworthy and fascinating is the amount of sort of adaptation that you have to go through to move CRISPR-Cas from the bacterial cell to being able to do genome editing. And that amount of adaptation is actually relatively small. You have to modify the protein in ways, you make a single guide, which of course is what Jennifer and Emmanuel figured out how to do. And then just put the enzyme into cells. And in one of the truly, miracle is not a word that scientists ever use. In one of the bigger surprises of certainly my professional life, this enzyme that has, in its natural state, has never seen chromatin. This enzyme has been able to work in pretty much any chromatinized setting that anybody has ever seen, anything from yeast to humans to actually living human beings, and everything in between. We don't really understand how that works, but it is noteworthy that this bacterial protein finds its way so comfortably through the nucleus of a human cell. This also is an important opportunity for me to mention that Cas9, the current clinically most widely used form, comes from Streptococcus pyogenes. There's one from Staphylococcus aureus, and work from several groups, including Matt Porteous, who's going to be speaking in a second, has really highlighted the fact that its immunogenicity, both either de novo or pre-existing immunity, is just essentially something that we have to be very mindful of when thinking of advancing any CRISPR-Cas-based approach to first in human, which is not to say that there are not reasonably thought through ways of how to mitigate that in a clinical setting. Now, I mentioned two pathways of break repair, and what I'd like to do is spend just a couple of minutes not only speaking about them, but highlighting what relevance they have specifically to the challenge of CF. This really beautiful schematic comes from one of the parents of genome editing, Dana Carroll, and the first process is putting the two ends of the DNA back together again. It's called non-homologous end joining, and HEJ. And when an engineered nuclease of any class induces such a double-strand break, in fact, most of the breaks are in fact repaired by non-homologous end joining. Having said that, it is possible to sort of subvert Mother Nature and drive a very formidable fraction of the repaired breaks to the other outcome, which is a lot more relevant. But one thing we've known for I suppose about 15 years or so, if not more, is that all engineered nucleases don't cleave once. They cleave multiple times before Mother Nature makes a mistake in connecting the two pieces of ends of DNA back together again, and thus creating small insertions or deletions. And this, of course, is useful to make a knockout. And these are 13-year-old data. You take a nuclease, you stick them into cells, and then you get individual single cell derived clones, which have small insertions and deletions precisely at the sides of the break. And of course, importantly, in this case, the central dogma plays itself out nicely, such that you start by Western with wild type cells, and then you end up with multiple single cell derived clones, which are genetically null and null at the protein level. So this frankly is a problem for cystic, because you don't want to induce an indel, because you are going to go from either a hypomorphic allele of CFTR or a null allele of CFTR to a really null allele of CFTR. And an enormous amount of work in the field has gone into understanding the interplay between these two break repair pathways and how to skew the outcome of the break repair. Bottom line, NHEJ is just a feature of life. Literally, it's a feature of life. And so this caveat emptor, or I guess editor beware. Fortunately, I would not be sitting here recording this talk if there wasn't another pathway, which is homology-directed repair. It typically uses actually a sister chromatid as a repair template. It's a unidirectional transfer of genetic information. And what's really beautiful about the sort of the canonical way of homology-directed repair is it is the case that the source molecule isn't even physically transferred into the chromosome. It's literally only the information is copied, but not the physical molecule. So Jim Haber, an extraordinary scientist who has done so much to unravel the mechanism of DSBR, even wrote a haiku about this pathway. He goes, "Chromosome broken awaits the sounds of strands pairing, preserving life's thread." It's just a lovely representation of what Mother Nature evolved this for. So Mother Nature did not evolve HDR for editors to introduce or repair mutations, but that, of course, is precisely what editors have done. So I need to mention that the word donor, I suppose in medicine, I'm not a physician, but in medicine, you think of a donor as somebody who donates an organ like a kidney or blood. In the genome editing slang, a donor is a repair template that is used to heal a double-strand break and thus subvert Mother Nature and transfer donor-specified new genetic information into the chromosome. And so what can you transfer? Oh, you can transfer all sorts of things. You can transfer a point mutation, and this is from work that my colleagues and I at Sangamo did in collaboration with Matt Porteous, what seems like literally the millennia ago. It seems a long time ago. But the foundational finding we came up with still holds, that you can use this approach to introduce a point mutation or repair a point mutation in transformed cells, which is what we showed at the time. And more recently, a lot of work by many groups has shown that you can actually make or remove point mutations with really good efficiency in dividing cells. And even more work has gone into trying to do something about the following problem, which is, I mentioned, and I'm going to just briefly go back to the previous slide with a haiku. The natural template for HDR, homology-directed repair, is the sister. And sisters don't exist in cells that are in G0 or G1 or mitotically quiescent. So for that simple reason, non-dividing cells are less HDR prone. Now, they're not completely HDR inert, but the bottom line is you can have a genome editing scheme where in dividing cells, whether primary or transformed, you can have a nuclease and a repair template that give you 80% overall editing, so 80% of the alleles are edited, and let's say 2/3 of them represent a precise correction or introduction of a mutation, and the rest are knockouts, small insertions or deletions. That's in dividing cells, and then you switch to a cell in G0, which is not tetraploid but diploid, which has no sister chromatids, of course, and suddenly the efficiency of the repair process plummets. So the overall editing is still high, but the repair by homology-directed mechanisms is actually unpleasantly low. So this audience needs no introduction to the fact that this creates an interesting and important issue as far as repairing CFTR mutations. And ultimately, as I'm going to say in a few minutes, only clinical practice will tell us what works and what doesn't, although non-human primate studies would also be important. But again, here's another feature of Mother Nature that we we have to collaborate with her, although there are interesting ways to subvert it, as you're about to see. So you can make a point mutation or repair it, which is great, but you can also subvert Mother Nature, and you can insert surprisingly large stretches of DNA. And one such result from 2007 is the demonstration that in transformed cells, you can take an 8 kb transgene and transfer it to a native gene with pretty remarkable efficiency. And when the really striking thing is, if you sequence this long thing, is you discover that it's been actually transferred to the chromosome very accurately. And that, again, has potential implications for genetic approaches to cystic, as you may hear later today. Okay, so I've talked to you about DNA repair, the two pathways, outcomes that you can observe in the context of genome editing, whether gene knockouts by creation of small insertions and deletions, introduction or removal of correction of point mutations, or introduction of larger things. So let me just walk you through briefly the sort of the canonical toolbox of genome editing, and actually spent quite a few minutes, well, not too many, talking about sort of the 2.0 toolbox. So the primary old school toolbox of editing is actually largely nuclease agnostic. My colleagues at Sangamo and I drew this for a review in 2010. And bottom line is you can do this with, we use zinc fingers, but you can use talents, Cas9, megatals, meganuclease, whatever else comes out. 2 pathways, and joining can create gene knockouts and make larger deletions as well as ligate tags. And homology-directed repair can repair mutations or introduce them, and introduce larger and larger and larger transgenes. I don't know what the upper limit is, but I certainly in my experience, 15, 20 kb can be done. So that's the original toolbox, folks. But Mother Nature is an amazing source of goodness, and scientists are incredibly creative. So I'm going to show you where this all went. And this is, let me see if I can remove my, I can't at time. I was hopeful to remove the little camera shot of myself, which is irrelevant here. It's a lovely slide that Megan Hochstrasser at the Innovative Genomics Institute put together. And what we basically see here is a progression of tools. So Jennifer's and Emmanuel's paper came out in 2012. It's a double-strand break with the outcomes you saw on the previous slide. And then, you know, the innovators kicked in, which is kind of extraordinary. David Liu came up with base editing, which is, it also uses Cas9. In fact, it uses Cas9 to create an RNA-DNA heteroduplex. But at that point, a different set of enzymes kicks in. And I don't want to diminish the magnitude of David's achievements by giving you like a 60-second Reader's Digest version, except to point out to one really fascinating thing. Base editing has been widely adopted for the repair or introduction of point mutations. And importantly, it's been recognized that base editing has formidable clinical promise. And there's obviously Beam Therapeutics, which focuses on it as a modality. But one recent development that I think is truly noteworthy for the CFTR community is Verve, which is a company started by Sek Katharison with a goal of reducing the burden of public health burden of cardiovascular disease, and with the primary target for now is to do a liver specific elimination of the expression of PCSK9. It is noteworthy that Verve chose base editing, and they licensed it from Beam to drive their desired outcome rather than plain vanilla cutting. So the general theme and lesson from this is the CFTR community has multiple sort of instantiations of a Cas-based or any other or a different nuclease approach, although base editing specifically does need a heteroduplex and an open loop, which something zinc fingers or TALs cannot create. So I'm going to skip through CRISPR-I, CRISPR-A, although I love them. And they're amazing. Jonathan Weissman and Luke Gilbert came up with that, and jump to prime editing, which is new. It's amazing. It's extraordinarily inventive. I'm on public record as having peer reviewed the paper. It went through pretty extensive peer review. But the only reason I bring this up is, I mean, it's just a marvel of how creative this is. Again, this is from David Liu's lab. They were inspired by actual naturally occurring mechanisms of retrotransposition. It involves yet a separate set of enzymatic functionalities, including a reverse transcriptase. The unique feature of prime editing is, and base editing also as well, is you do not need to provide a separate repair template in order to create or repair a point mutation with prime editing. So the donor is not a separate molecular thing. It is in fact carried by the prime editor itself. And this sort of all-in-one capability of this machine was, to me, when I was reviewing the manuscript, it was just like literally my eyes glazed over with joy, because a co-delivery of a repair template is doable, and many people have done this very well, including ultimately in clinical settings. But this fundamental idea that you can have an all-or-one machine that goes into the cell, goes into the nucleus and carries with it the instructions to repair the break to a desired genetic outcome. That's pretty thrilling. And Prime Medicine, again, David started the company, is going to take this to first in human. I don't know what they're going to do, but the CF community really needs to think about putting this to use, and I'm sure already is, with caveats that I'm going to address at the last slide. So in closing, the organizers have asked me to give a little summary of a previous experience in first in clinic. The first in human trial was done by Carl June and Bruce Levine at Penn with Sangamo, my colleagues at Sangamo, Dale Lando, Mike Holmes at Rebar, and yours truly developed the approach. And but then editing entered the clinic with TAL effectors for cancer. Then zinc finger trials began for sickle and thal, and then in vivo for lysosomal storage diseases and hemophilia B. Then Editus put eye editing into first in human, and they also have an open clinical trial for sickle and thal. CRISPR Therapeutics have an open clinical trial for sickle and thal with some spectacular successes in one subject with sickle and one subject with thal. And I'm probably missing a few there. Carl June did a very nice trial at Penn for cancer with editing. Selectus is in the clinic for a bunch of cancer indications. The central lesson I have for anyone who is considering clinical editing is the only way to find out how to edit people is to edit people. There is no preclinical model that will conclusively de-risk your approach from a safety perspective, and there certainly is no preclinical model that will de-risk your approach from an efficacy perspective. And we have shining examples of the latter, gene therapy trials for thalassemia, the one at Memorial Sloan Kettering versus the one done by Bluebird. Markedly different clinical outcomes, even though it's the same approach, a lentivirus carrying a beta globin transgene. For the more recent work on the hemoglobinopathies, two companies are in clinical trials, Sangamo and CRISPR Therapeutics. They're editing the same base pair of the human genome. One is using zinc fingers, of course, CRISPR is using Cas. The CRISPR data look great, the Sangamo data less so. This basically says one thing and one thing only. You have to take your favorite approach to first in human sooner rather than later. There is no preclinical study you can do to convince yourself that it's completely safe, it's going to work. You just have to bite the bullet and do it. Three things matter in cell and gene therapy, and that's delivery, delivery, and delivery. That's a truism, it's a foundational fact. I think considerations of ex vivo approaches for cystic are important to think about, and we might hear more about this in a second. As far as delivery, don't underestimate the fact that iterative technology improvements do add up. They do. So things that were technically completely unimaginable a decade ago are now relatively straightforward. And if right now, as you think about the size of the target organ and percentage repair and all of that good stuff, there insurmountable hurdles may seem to exist. They will be in fact surmounted. Honestly, if I had a ruble for every time somebody asks me whether prime editing is better than talents, I would be an oligarch. The street has this obsession of who is better, and what is impossible to communicate to them, but is possible to communicate to scientists and clinicians who are in the audience. It's not a zero-sum game. More shots on goal is better. Again, back to my favorite hemoglobinopathies. I mean, there's multiple clinical trials with gene therapy, plain vanilla, including both for transgenesis of the beta globin and for fetal globin elevation. There are multiple clinical trials for elevation of fetal by editing. There's going to be a trial from Stanford. Matt Porteous and Maria Grazzaron-Carollo are working on that to repair the sickle mutation, and the IGI is working on that as well. And it's great, because ultimately, as I said, the only way to find out how to edit people better is to edit people. So I really don't see any need to have a zero-sum game type debate about prime editing or bust for cystic or what have you. I think as long as the approach passes FDA guided and defined preclinical safety and efficacy. And as long as the efficacy data have produce a target product profile that says that there is a there, and if you can enroll the trial and do this under all the appropriate regulatory framework, you should just do it. And the last thing I'll say, and this is, I suspect that I'm preaching to a lot of card-carrying members of this choir, is I think we live in a time where it's now becoming clear that no specific individual effort can succeed in an endeavor so complex. By that I mean, it really is not enough to build a better editor. It's not enough to build a better delivery system. It's not enough to build a better animal model for any of those things. You kind of have to build them all together and put them together in order to get to first in human. And I'm saying a truism, and any biotech is actually doing precisely that. And that's of course called vertical integration. And one of the things that I find striking, and this is the last thing I will say, is the extent to which there has been really a lot of fantastic work advancing editing to first in human in academia, Penn, Stanford. We at the IGI are hoping to head to clinic, and I wouldn't be surprised. Oh, so I'm sorry, Seattle, Children's Boston. Again, I'm probably missing folks, but my big picture point is I think academia offers a unique framework, especially in the context of, if we're leading forward-looking clinicians, to do some serious good. And I will close, in fact, just to highlight the fact that, we don't just talk the talk at the Innovative Genomics Institute, but we walk the walk. I'm very grateful to two sets of people. First, Ross Wilson, my colleague at the IGI, who pioneered this whole thing and came up with the idea and wrote the grant with some input from yours truly, and then for the Cystic Fibrosis Foundation for supporting it. And again, my goal in sharing this is not to sort of say, look at how good we are, ask us again in 2 years when we're done, how well this worked, but simply to point out this notion of vertical integration. So, we've collated people who think about how to engineer enzymes to drive outcomes with people who think super hard about how to deliver them, specifically for cystic, with people who build really dedicated, sophisticated, bespoke ex vivo models, as well as in vivo models that allow really robust, as robust as it gets, preclinical safety and efficacy testing. I'm very excited about this because, obviously industry is essential to advance these and ultimately commercialize these therapies. But, and this goes back to my very, very first point, if the example of CRISPR-Cas and Jennifer's and Emmanuel's discovery, and there's hundreds of people whose work contributed to ultimately their success, for example, a decade plus of work on bacterial adaptive immune systems that are of CRISPR, of the CRISPR type. Fundamental innovation, which is intrinsic to academic settings, can be quite enabling for very applied things. And I think on balance, I don't want to seem Pollyannish or naive. My personal sense of overall optimism that there is going to be a first in human trial for editing of some flavor for cystic in the next 5 years is actually quite high. How well it's going to go, I don't know. But ultimately, if the 30-year history of cell and gene therapy teaches us anything, is that iterative improvements and the collective wisdom of the field, industry and academia, ultimately can solve a very formidable fraction of the challenges. Thank you. I appreciate the opportunity to speak with you today.